So good morning everybody, and thanks to the organizer for the opportunity to show my data. These are my disclosure. For the ones which are not familiar, during my PhD studies I'm looking at the purinergic receptor regulation of immune cells, and what I observe is the role of molecules such as ATP that when it is released normally by cells which are activated or during inflammation can act of purinergic receptor. The ones we were interested in are ionotropic receptor, and in particular the isoform 7. It is really important because it is the only one which has no effect when ATP is in low dose in the extracellular space, but when ATP increase, then you got ionotropic function of the receptor that allows the entry of calcium, for example, and it is linked to innate cell activation, inflammasome, and so on. To us was really interesting to see what happen when ATP is really increased in this extracellular space, which means, for example, chronic inflammation. At this point, the receptor, it is the only one of the family that can opening a pore and allows the entry of big molecule, and this is connected to apoptotic pathway of the cells. We previously shown that the knockout mouse for this receptor display an important phenotype without any kind of treatment in the gut, where a subset of T cells, which are TFH cells, are increased and determine this biotic condition. But the number of TFH cells, actually, it is really a big question in the field of normal and autoimmune disease, because you don't really care to have the more the better, because you want to stay in the threshold to be safe. So we wonder if in our case, in autoimmune context, the ATP release and chronic inflammation could have somehow a role in controlling TFH cells. And I was really focused in study the SLE as a model where you break the immune tolerance and you have chronic inflammation. And to do this, I performed this kind of lupus-like disease in murine model by injecting the mouse with crystine and by looking at the late stage of adaptive immune cell activation. What I observed was that basically my P2X7 knockout mice were more susceptible to cell death and also to organ damage, because I found increased splenomegaly and lipogranuloma formation, and I also correlate the end organ damage of the kidney with the increased presence of abnormal glomeruli and the worsened function in terms of level of proteinuria, and these were all correlate also to immunocomplex deposition. I checked the serological feature of my mouse, and I found also a polyclonal expansion of IgG, which were of autoreactive kind of IgG circulating in the sera. I screened the kind of pattern, and I found that the mouse were affected by more homogeneous kind of staining, which means they were really more similar to what is a lupus-like disease in mouse. And I also performed a more sensitive screen by adapting a human kit for the searching of 9 known autoantigen, and I basically found that this mouse expands autoreactive IgG in the sera. We were interested in immune cell activation, as I told you, and what I observed was that at the end of the experiment, just by the induction of crystin injection, the mouse were maintained a constantly activated state of CD4 T cells, and these cells were polarized to type 1 interferon gamma, which is actually related to many model of lupus in mice and also to increase kidney damage. And to us was really interesting to see that specifically we got an increase of T follicular helper cells, which were also in the case of autoimmune contests not regulated more if the P2X7 receptor was deleted. And this increased presence of TFH cells will correlate with sustained germinal center B cell activation. So to be sure that the mechanism was T cell dependent and intrinsic, we tried to adopt transfer T cells. When we did this with transgenic wild-type or knockout T cells and foreign antigens such as ovalbumin, we didn't find any difference in terms of expansion and follicular helper capacity. But when we reproduce the same experiment with polyclonal T cells and by mimic self-antigen with pristine injection, we found during time an increase of TFH cells which were deleted of P2X7 receptor, meaning that the control was really dependent on T cell activation and expansion. And we were also really interested in translate the data from mouse to human because of the relevance and because pathological role for TFH cell are well described. Nowadays, for example, these recent papers show that in lupus patient and not in RA, you got expansion of these TFH cells that in this case they consider as PD-1 positive T cells. And we have the opportunity to collaborate with Professor Meroni, which helped us to collecting the donors and to compare SLE with primary antiphospholipidic syndrome. We were really lucky because we confirmed exactly the data. In lupus patients, we got increase of TFH cells, which were not increase in primary antiphospholipidic syndrome. So we had the internal control of another autoimmune disease where TFH cells is not to play a role. And because we knew from the past that effector T cells also in human are regulated by P2X7 receptor, we checked the mRNA level and we found a significant decrease in lupus patients which were not significant in the primary antiphospholipidic syndrome. To be sure that there was a correlation with the function, we perform this kind of flow cytometric assay where the cells are not normally responding to big molecules such as EOPRO, but just when you add a higher dose of the agonist, you got responding cells, as you can see from the dot plot, where the cells start to be positive for the EOPRO. And we did statistically the analysis for healthy versus lupus and primary antiphospholipidic, and what we found was in line with the expansion of these cells. So basically, it seems that lupus patients do not really respond to apoptotic signals which are mediated by tau receptors. So to summarize, what I show you is that when you induce lupus-like disease in mouse and you don't have the P2X7 receptor, you have worsening of clinical and histopathological outcomes. This expansion of specifically follicular helper T cells is a mimic of what happened in lupus patients, and it is not the case for primary antiphospholipidic syndrome. So maybe the receptor could be not only telling us a mechanism of control, but probably a pharmacological target for lupus patients. And the last slide, I want to thank my group in Bellinzona, the group of Fabio Grassi and all the collaborators around, and especially Pierluigi Meroni and the group which collect the donors and help us also in discussing all the blood donors, which allows the study of this kind of study. And thank you for your attention.